Me is a great honor to have taken you as a student, Kaylin, in light of what happened with Mark. So I am pretty sure he would be extremely proud of you today if he could be with us because you did grow to the type of professional that he would be preparing you to be.
Woohoo, he is with us.
Excellent, thank you. Okay. Oh, bugger, all right. Okay, there we go, okay, it moved. All right, so kind of the background and the basis of what the goal was of this project is imaging is gaining a lot of traction in controlled environment agriculture because it's something that you can do at scale very easily, where it's something fast. Easy is debatable, but once it's set up, it becomes so. And then the beauty of it is that it's non-invasive and non-destructive. So you can do it repeatedly on the same plants as they develop. And you can use it to quantify an assortment of things. So you get your canopy size, you've got your pigment coloration, your concentration, sorry, and then stress symptoms, which are often manifest in both pigment concentrations and in the physical manifestation of the plant. And the beautiful thing about using an imaging system rather than just a sensor per se is you can get some spatial information, so you can kind of piece together what's happening and whether or not you've got localized stuff and start building from there. So where we started out with the top view phenotyping system. The hardware was robust if poorly thought through in a couple of regards, where it provided us with 8 channels that we could work with for our imaging, and it generated monochrome images, and so we could put those together with ImageMath to make what we wanted out of them. But this had a pretty core set of weaknesses, where it's expensive, which really reduces your ability to use them at scale. And the analysis was not automated and it was not customizable, where one of the core tenets of why we had to do a lot of our own post-processing is I believe the 870 nanometer channel. The images would get shifted, or it was kind of rather because there was a lens, and the image would get warped and shifted because the lens was never in there true and straight, that they tried to use to help refocus the image. And that was kind of catastrophic. And I don't know if Chung-Hyun talked to you, Mark, about that much, but I think that was a lot of his programming pain, was trying to warp it back so it was in the same place to be able to do reasonable image math. Thankfully that predated me. So this led to kind of the objective of the project that Dr. Van Ursel laid out for me when I came out here, was working with the hardware that Benjamin had put together and the code that Chung-Hyun had worked on to kind of put everything together to make a functional image collection system that would analyze the images. And then the goal of the studies that I ran was to validate that capability to be able to pick up the differences in plant canopies. And so to do that, I used fertilizer applications and pH of the substrate to drive those changes in the canopy. So this is what we ended up putting together. So if you look in the first picture, you can see the smaller camera module, that's the original, with the lens peeking through the plexiglass plate over a plant. The second image is one of the big pieces that I added, was the chlorophyll fluorescence imaging module, which is just a hobby servo and a whole bunch of snazzy 3D printed parts on that poor printer. And then putting that together with the requisite code to put that servo where it needed to be. Because the thing that really messed up the top view system is they used an incredibly cheap stepper motor and it was not repeatable. So the filter wheel would be all over the place. And that didn't help anything. And then another one of the core things that I worked on is Benjamin did a darn good job putting this together, but over the time and the experience we had using it, it turned out that these nice LED clips that can be seen in these pictures that clip onto the end of the lines ended up having horrible connection issues by the time I got to it, and I'd have I'd have individual strips go down on me when I was in the process of trying to collect images, and so the entire thing got rewired and kind of streamlined a little bit, which was the benefit of time. So it was about $600 plus the labor to get this all put together. And though there's a whole cost breakdown on a following slide, and that cost is not adjusted for inflation, I don't think. And it depends on when you were purchasing your Raspberry Pi, whether or not that's a $600 or $1,000 price tag. But a lot of the key to this, so there is a lot of there was a fair amount of labor associated with this, which I'm sure Benjamin and Chung-Hyun would second, because it was a lot of figuring out, which was a lot of Benjamin had to figure out how to run all the wires, and Chung-Hyun had to get it all to play nice. And so, but at the end, it works. The Raspberry Pi processes those images in real time, and you can tweak whatever spectra you want out of it. There's another graduate student. So I believe Mark Irdakunda was talking about putting another index in there at one point for, I don't remember who it was, but I'm talking about putting another index in it at one point with him, but I don't know that that ever happened. But by the time it's all done assembled, you just throw more LEDs in there, hook up a relay, and you just run the software, you just run the code loop one more time. The education was really valuable. I think that I assume that's put Benjamin a long way forward, and it certainly forced Chung-Hyun and I to up our Python game considerably. So the education, I don't know if that's a cost or a benefit, but that can be debated in both ways. So here is that breakdown of the pricing. So I don't know how far we want to get into that, but that's roughly what we came up with. And a lot of this is substitutable. There aren't a whole lot of these that are straight requirements. So it needs to be a specific long pass filter, and it needs to be the specific camera, but other than that, it's pretty free and loose, which is one of the benefits to the system. So the way that this system works, which you guys should mostly be associated with, or that you should mostly be familiar with. So that's not a great sign. Okay, there we go. This is an older video because the Raspberry Pi is still in there. One of the things we did is we moved the Raspberry Pi outside to help with some of the light homogeneity and ease of modification. So it flashes the lights, takes the monochrome images, and then we can start doing the fun stuff, which is the image math, where we start getting useful things out of it. So there was some coding where there was some optimization, and the program it turned out the IDE that we were using had a catastrophic memory leak. And so we were all living in pain. And it took me a lot of images to realize that my life would be a lot easier if I switched IDs and tuned a couple things up. So that's all. So this was all for the analysis, the post-processing analysis of the images from the commercial system, which is a lot of what drove the Raspberry Pi there.
May I jump in very briefly? You found that out with memory leak and ugly things like that on your own?
Yeah, well, not before I upped the RAM in my laptop to like 48 gigabytes to try to buy myself enough time to analyze enough images to be meaningful. And then I figured out that was what happened.
Nevertheless, that's a feat. No, that's impressive. Go on. Sorry for the interruption. I was just, well.
No problem. Thank you. All right. And so the the primary index that we were looking into, because it has a lot of strength in plant-specific fields, is NDVI, where it effectively is the greenness effect of the pigmentation, where in the top set of images, which I did not align as precisely as I thought, you can see where's the mouse? There's the mouse. You can see where this leaf is dying in the back, and you've got some pretty bad chlorosis. And then you've got a tangible difference between here and here, but in a field full of plants, that's hard to pick out when you're not against a black background. But in the NDVI, it really shows, because you can see the new leaf is hard to pick out in the color image. But here you can see there's the new leaf over the older leaf that's beginning to lose its functionality. And then this is a high fertilizer plant, and it's pretty homogeneous and it's pretty happy. And you can see that in the distribution of pixels on the histogram. That's what these things are called. So where you can see also that the bottom plant is a lot larger because at 0, there's the spike for all of the background pixels. And you can see that's a lot larger in here. So that's a representation of the size of it's a representation of the amount of plant area. Then you can also see that it's a much wider peak. And so you have a much larger difference because you've got these lower NDVI leaves and the higher NDVI leaves. And then compared to the healthier plant, which has a much tighter spike, as all of the biomass is much more homogeneous. So the chapter 2 was all about putting that imaging system together, the constituent elements, what we did differently from, in some ways, other people who were doing similar work. A lot of people that I looked at, they will just, because the Raspberry Pi cameras are so cheap, they'll just start stacking cameras and put a filter in front of each camera, which feels like more work than it ought to be, which is why it's the one camera in the black tent. So it put it all together and then now we're into the supporting data. So for the fertilizer study, we chose fertilizer because it has a strong effect on how plants react. As shown in the previous picture where there's the huge difference in the biomass, and I believe that was a lettuce, and then also in the canopy coloring. And so the objective that we laid out for this chapter and study was making sure that, since we took these images with the commercial system, which really helped us understand where the weaknesses were and helped expedite the development of the in-house system some more. And it was also fine-tuning that analysis code that we ran on the computer that then we rolled straight across into the Raspberry Pi code. So both of these trials were in Petunia. Well, this trial, all of my trials were in Petunia. So putting different rates of fertilizer and then irrigating with just water. And then putting controlled release fertilizer on. That was one of the effects that if I were doing this again, I would like to change, because we did a longer term controlled release fertilizer that's supposed to have a lifetime of something like 3 to 6 months in the soil. And we were growing for at most 2 months, maybe 2.5. And as such, we didn't get the full effects of that fertilizer. And so some of our responses were a little bit muted because of that. And then these were grown in peat and then 8 plants per treatment per cultivar in here in 3 different cultivars. And now we get into the graphs. So I know I read your comment about the biomass against the canopy size, and I'm going to change it. It's not changed in here, but it's going to happen. Didn't feel like fighting with Sigma plot on the airplane. So here, these are all kind of tied together, the size measurements. So we've got projected canopy size as a function of the fertilizer, and that's on the wrong screen, as a function of the fertilizer rate. The dry weight is a function of the fertilizer rate. And then the biomass, the dried biomass or dry weight, I'll need to change that too. And then the projected canopy size. And so these all, so as we kind of, as we were hoping and anticipated, as we added more fertilizer, we got bigger plants with very consistent results. And then as the projected canopy size is just a base measurement of the plant size, but 2D projection, it should also track well with the fertilizer rate, and it should be correlated very strongly with the biomass, which all worked out real fine, as advertised, I suppose might be the best way to put it. And then we start getting into the fun stuff where we're looking at nitrogen against the fertilizer rate and the NDVI against the fertilizer rate, and those also both have those kind of positive trends connected with the increased rate of fertilizer application, and then tying the NDVI with the nitrogen. That was a little funny. I don't know why it's curved like that. An opportunity for further research and potentially a debate with somebody who is more molecular or more of the molecular physiology and might have insights as to the connection between the nitrogen concentration and the NDVI average, or perhaps why the uptake was well, actually, I should have put that on a log scale in hindsight. Might change, anyway, might change that later because the log scale actually would make more sense in that regard. So, and that all kind of checked out how we anticipated it to. Got some interesting outliers that I don't have good answers for. But on average, everything kind of dropped where we expected it to. And then tying together the projected canopy size and the nitrogen concentration, we expected a slightly stronger relationship, or at least I did, between the nitrogen concentration and the canopy size. It checked out and it's significant, but I suppose I expected a stronger response from these a little bit. And then the projected canopy size against the NDVI, which was just kind of, that was to try to get a little bit more insight in an instance where a lot of commercial growers will have a ton of plants per tray and there will be a huge amount of overlap. So they can get an NDVI of the area of the whole tray, but they can't necessarily get individual plant sizes. And in floriculture or decorative horticulture stuff, they're often shooting for a plant size for marketability. So if you can take an NDVI of the tray and have some understanding of how your sizes are doing to some extent without having to segment all of the plants apart and do all of the fancy segmentation to separate overlapping plants. That'd be a quick and dirty way to hopefully get away with it. And it's significant, though I don't know how much I would predict on Super Cascade Red or Carpet Blue as opposed to Wave Purple, if I were using that as an effective tool in the field. But it's a potential opportunity if Renito wants to start stacking plants and seeing if he can predict size with NDVI in the future. And so the goal of this was to verify that the code that was put together was effective and that we could then port it over to our hardware that we put together. We could pick out the differences between the treatments. We found significance in a lot of it. And I would consider that to be successful and that we completed the objective for that trial to where our code fit what we needed it to do. So now on to chapter 4. In hindsight, now that I've done a lot more reading, pH in particular in petunias was not a great choice because as it turns out, petunias don't actually care a whole lot about the pH of the soil, which might be why they're all over the face of the earth. So we were trying to induce canopy variation by using those different pH rates because it controls a lot of the micronutrient availability, for a lot of plants and in a lot of situations. So trying to do that and then being able to pick out those differences using the imaging system. So it's a very similar layout, but this time we used a fertigation solution and a pH amended fertigation solution. And same experimental design and the same 3 cultivars, though growing in rock wool to try to mitigate any kind of media effects. Because the 2 trials I tried to run in peat, I didn't get any kind of meaningful pH effect from the fertigation solution, which was kind of a bummer. So we've got the effects of the pH on the phosphorus and manganese concentrations, which are pretty vigorous. And then we get down into the NDVI for the treatments. And we, on paper, did not find significance in any of these, though under consultation from my statistician, the outliers in Super Cascade Red were exciting enough that this could be considered significant. With the deviations here are pretty wild. So with that being said, we then pivoted over to chlorophyll fluorescence intensity because as part of the chlorophyll fluorescence image thresholding to segment the plant from the background, that we also got that chlorophyll fluorescence index, or just the chlorophyll fluorescence intensity. And so we started plotting that against the pH and against the nutrient levels because both manganese and phosphorus in papers that I read had effects on chlorophyll, on the quantity of chlorophyll and the intensity thereof. And so rolled into the chlorophyll fluorescence intensity as that was another one of the tools built into that imaging system that we can easily leverage. And found some significance here. Super Cascade Red, where it wasn't as susceptible in the previous trial to some of the fertilizer effects, was considerably more susceptible to the pH effects. And so the CFI, you kind of see that you're starting to have issues. So in an integrated imaging system, if your you can use all of the different spectra to pick out when you've got a problem. See if your NDVI doesn't show something, but your CFI does. The goal, in my understanding, is to make available a tool where you can identify when something's suboptimal to rectify that, and using whatever spectra you've got gives you additional insight where you may not otherwise have it if you have somebody going out there with the naked eye, because I personally can't see chlorophyll fluorescence with my naked eye, let alone quantify it. So I consider that a useful tool, but I'm also a sucker for a good measurement. And so then here we are into the projected canopy size data. This stuff didn't track so good. These may be some of the highest p-values I've ever seen in my life. So the pH didn't do much to the canopy size, which is probably why everybody and their dog plants petunias, because they don't care what you plant them in. So that did find literature that said, yeah, the petunias just don't care. They're more or less pH inert, which was a little bit frustrating for this trial, but it was interesting to see the nuances in the chlorophyll fluorescence. So the end goal of this was to develop a tool that could pick out changes in the NDVI, in the chlorophyll fluorescence intensity, and just in the plants in general. So we didn't do a good job of inducing differences in the plants, but what differences we managed to induce, the imaging system, I think, did a good job of characterizing those, which was the end goal of putting that together, was a tool that could detect and quantify those things for future research opportunities. So these are what my objectives were: to put everything together and to make sure that it worked effectively. And the conclusion that I came down to is it does work, though we're a little bit limited in its current setup for the cultivars or the crops that we're working with. Because past a certain point, the petunias were getting too big for unrow. And there are a lot of crops that are too big. And based on the way that it's designed, it's not particularly effective during daytime in greenhouse applications for looking at long run stuff. It has to be in the dark. And so it worked fairly well. I think it was fun to see the different cultivar specific reactions to the different treatments that were applied. And the things I'd probably do again, I'd rerun everything again. Though, for example, in the fertilizer trials, I'd use not a slow release fertilizer. And I probably wouldn't do the wave purple again, because that plant, it just kind of goes and just makes a carpet. And the other ones have a much more vertical canopy structure. So it limits the ability of the 2D plant canopy area to give you an idea of the plant size, but it does allow you to image much more consistently without having issues with imaging system space. I think it'd be fun to experiment with a couple new indices, because they're not hard to add to this, which was a lot of the design goal. It would probably take me worst case a day to add a new index to this system. Best case an hour. And that includes the LEDs and stuff. And then the other thing that I learned was the value of having multiple cultivars in there. Because if we'd only looked at one of those cultivars, then would not have gotten a very good idea of the picture. And then other things that would be fun to try. If we're just purely looking at fun, it'd be great to hook it up with a conveyor belt. But in terms of other analysis options, subsegmenting the plant canopy area and then taking averages of each of those subsections. And so that was something that I believe a number of WIN students looked at. And then Jeff, whose last name I have forgotten, who was here for a little while. Jeff Weaver. Yep. He started doing a little bit of that as well, but it would be interesting to continue some of that work with a couple more plants and develop that a little bit more.
So the rerunning is the main reason why you were here today, so you brought him back to, continue with that research?
Yeah, next time I'm automating the pH controls. I was under there 3 and sometimes 4 times a week with a pipette under the table, dosing those tubs and stuff. Next time,
I need to
automatic.
add a point though.
Uh-oh.
So his brother and cousin were busy.
yeah.
6 months later, they had something that would have done it for him automatically and made his life much less interesting.
Well, and kind of as a proof of concept for this, I sent my I sent actually that same brother the designs and the code that we'd put together. And in a month, he had built a duplicate system in Washington State, but he used a different camera library. And Mark, I don't know if you remember, we were having those weird spikes in the histograms when we were trying to
Yes.
segment the. He used a different camera library and the spikes are gone.
Probably too much side, that's probably too much side, anyway, funny how
Anyway.
things work.
So, which was kind of the goal, is that other people can build it quickly and easily and hopefully make improvements. So what took us 2.5 years, took somebody else with the plans a month, which was kind of the goal, and then.
That's the
Oh,
point.
science.
And it's a little
Yeah.
better, yep, so
Yay.
the goal is to hopefully give people the foundation they need to build the imaging system to collect the data that they want. So. I'd like to acknowledge Mark, who put up with me and dragged me across the continent for this, and then Renito for adopting me. And then to you, Mark and Lynn, for filling out my committee, and Mark for helping me figure out what all my technical problems were at the beginning. And Lynn for helping make sure that that I was barking up the right tree. And then all of the lab mates who helped me with everything. And actually it was Peyton who was like, oh, I have this giant harvest, that induced me to rewire the entire thing instead of just dealing with the loose wires from my data collection. And then my family for putting up with all of the frustration and edits that they've put through. So funding came from the Fred C. Gluckner Foundation, the American Floral Endowment, and the Horticulture Research Institute. And from the Horticulture Department, I believe, as well. So,
And your grandpa.
not on the funding on this
Oh,
one.
no,
including-
and did just on
Yeah,
the family.
Oh, yeah,
tree.
no, but, yeah, no, Grandpa sat through a lot of technical questions too. All right, and then I think that's the lot, and now we get to the interrogation section.
Wonderful. Thank you very much, Kaylin.
Mhm.
Appreciate it. So if you want, you can perhaps stop the recording.
right.
Yeah, there you go, more.